Collective cell motion is indispensable for several developmental and physiological processes within an organism. As an example, here we see movements of endothelial cells, cells that form the blood vessel network within an embryo. Some nuclei are traced, and the trajectories reveal a locally ordered movement pattern. To understand how such cells can organize their movement, we performed an experiment with cell cultures.
Cells were grown on a rigid plastic tissue culture substrate and seeded in patches in which the cell density was very high. We used an immortalized human keratinocyte cell line named Hecate, which we could easily culture and manipulate in a standard cell culture lab. The cell cultures were grown in DMAM culture medium supplemented with bovine serum in culture plates kept in a standard incubator, providing for optimal temperature, humidity, and atmosphere. Cell movements within cultures were recorded by an automated microscopy setup, which monitored parallel cultures for several hours up to a couple of days. We placed the cell culture plate into a transparent mini incubator mounted on the powered stage of an inverted microscope. The stage-top incubator provided for standard cell culturing conditions. We selected the fields of view manually and stored their positions in the computer. The time-lapse program moved the stage into the stored positions every 10 minutes and captured single images, which were then used for time-lapse videos or motion analysis. This time-lapse recording shows the dynamics within a high-density patch of epithelial cells. Cells are cohesive, slowly expand the patch while participating in a vigorous, turbulent-like flow within the bulk of the monolayer. The spontaneous cell flow is sensitive to perturbations of cell-cell adhesion. If a calcium chelator agent disrupts adhesion, the co-moving domains break up into smaller groups, and relative movement between the cells, the shear, increases.
This animation shows the changes we assume take place at cell adhesion complexes when cells move in streams. The yellow bars indicate cell-cell adhesion sites of the gray cell. As cells move against each other, some adhesion complexes must break while new ones must form. As these processes take time, adhesion complexes separating cells adapt slowly. In particular, we expect less adhesion sites present in the front of a moving cell. As a simplified model of adaptive cell-cell adhesions, we consolidate the spatially distributed adhesion sites into a single spring-like connection linking adjacent cells. Like the dynamics of distributed adhesion complexes, the proposed dynamics results in an asymmetric distribution of link directions. This dynamics was implemented in a self-propelled cellular bots model. In this movie, yellow cells are self-propelled while the red ones are diffusive. The left panel shows the traditional dynamics, while the one on the right is a simulation with the proposed link dynamics acting between cells of the same color. Simulations performed with confluent monolayers reveal an intermittent, locally ordered dynamics. The width of co-moving streams can be tuned by the parameters characterizing cell adhesion dynamics. In general, the streams are broader if the cell adhesion links are slower to form or have a longer lifetime.
From the velocity fields of both experiments and simulations, we calculated the average velocity field surrounding the moving cells. The flow field around control cells revealed larger correlations when compared to the more anisotropic field surrounding treated cells. Simulations show that this anisotropy is controlled by the strength of adhesion between cells. Stronger links result in more isotropic flows.